Hello, everyone. It's wonderful to be here. I wish I could say with you, but let us hope for next year. I wanted to share with you some of the exciting work that we've been doing to try to curb viral diseases. And what we're trying to do is to provide a rapid solution for these infections. Now, you see, the challenge that we're trying to overcome is that there are no platforms to generate antiviral drugs. Every antiviral drug that was devised necessitated a unique strategy. And that's the reason why it takes us so much time in order to combat every new pandemic. So what is our solution? Our solution is to look at the ingenuity and amazing resourcefulness of bacteria, the most amazing creatures on this planet. And why do I say these accolades? Because bacteria are able to resist nearly every external threat that you provide them. They're able to live in extreme temperatures, extreme salinity and acidity. They're able to combat and develop resistance against any chemical that you throw at them, i.e. antibiotic resistance, and they're also able to withstand radioactivity. But the problem is that bacteria and viruses reside in parallel universes. One does not affect the other. Viruses are not a threat to bacteria, so why would the bacteria generate agents that harm these viruses? Now, the solution that we came up with is ion channels. So what are ion channels? They're components that are found in every organism that enable it to generate a safe environment for itself, to regulate its internal salinity and acidity. Interestingly, many viruses also have these channels that they use to regulate their environment, and without that, they're unable to conduct their infectious cycle. As a family, ion channels are fantastic drug targets, and in fact, many drugs that we take against high blood pressure and numerous other indications block channels. Now, the best thing about ion channels, and that's the trick of our technology, is when you express viral ion channels in bacteria, they actually impact bacterial viability. So you see, for the first time in evolutions, bacteria finally have a reason to combat the virus. And the solutions that bacteria can come up with may be beneficial for us. So the task at hand is relatively simple. The first thing that we need to do is to identify and confirm ion channels in the particular virus that we're interested in. We then need to screen for blockers. And finally, bacteria also enable you to assess the resistance potential against any new antiviral drug that you generate. So what's our current progress? Obviously, everyone, including ourselves, is interested in COVID-19. And what we've already shown, using all of our bacterial assays, is that the virus contains two channels. SARS-CoV-2 contains two channels, and for us, a channel equals a target that we can block. We've screened roughly 3,000 approved for human use drugs against them, and we were able to identify close to 8 inhibitors for each one of these channels. The most exciting part of our work came when we were able to show that these compounds that we found inhibit the virus itself. This obviously necessitated a biosafety level 3 lab, but once again, we were very fortunate to have the Friends of the Hebrew University provide support and to be able to stock up our lab and to make it as functional as we require. The next phase, which we are currently starting right now, are animal trials, and we are very, very hopeful that those will prove that the compounds that we found are actually anti-COVID agents. And to give you an example, this is the only data slide that I'm going to show you. An example of what this looks like is to actually check for viability of cells as a function of viral infestivity. So you can see on the left, you have cells that are grown for 48 hours, and on the right, the column that says no drug, you can see that close to half of them die because of viral infection. But when you give them small concentrations of the drug that we have found, in this instance darapladib, you can see that it prevents viral-driven death almost entirely. And I can think of no better example of antiviral efficacy. Because we're talking about a platform, I can prove that because we've already identified channels in other important pathogens such as Zika, dengue, West Nile virus, and even hepatitis B. We've identified blockers against each and every one of those, and currently we are assessing the efficacy of these blockers against the whole virus. So to sum it up quickly, we are very hopeful. We're hopeful that we were able to come up with a platform that can respond rapidly to viral threats. And as an example, one of these compounds, and in fact 2 of them, are potent anti-COVID agents. So let's hope that the next time that we meet, I'll be able to tell you about our success in person.